Lovely, thank you. So in full disclosure to begin with, I have been asked to give demos, so I will do, but I'll use FSL to do that, and I get some royalties from it, although it is free for academic use, so hopefully that doesn't affect you here. So let's think about ASL post-processing now you've got the data. So we'll do our demos with the Basel toolset from FSL, and you'll see as we go what that looks like. So the way I want to approach the idea of analysis and post-processing for ASL is really to assume that you've pressed the ASL button on your scanner and you've got some images and you've gone, okay, what have I now got here? And so the question is, do you want to do something very simple and easy, or are you really trying to aim for what we can do with ASL, which is to get absolute perfusion measurements? So going right the way back to what we've been thinking about this morning, can we measure perfusion in absolute units to say something about the physiology of the brain or whatever organ we're interested in? So what we want to do is something like, what have I got? What do I want? And then what I'm going to try and answer is, what should I do? What should you do with the data? For future reference, I'm going to put some command line instructions across the bottom. If you see the blue box, feel free to ignore it. Because the talk's recorded, you can come back to that later, but it's mainly just so you can see what I have done if you ever want to try and do it yourself. So what we're going to do is we're going to keep it simple to begin with, then we'll think a bit more about quantification as we go through, and then at the end we'll come back to thinking what happens if this is part of a larger group study, and what else might you want to consider if you're going to do really robust or more accurate and more precise quantification. So let's keep it simple to begin with. We've said ASL has the big feature of ASL is a control image and a label image. So what you've got is some ASL data. You've probably got a series of control and label images, and you're just trying to get a perfusion image out. And so as we've seen, the key step, the first thing you always do is label control subtraction. And that gives you a perfusion weighted image, and if you just take a single pair of label control images, you'll get a very noisy perfusion weighted image. But the chances are you will run this a number of times over a course of a couple of minutes. You'll have a number of images, and you'll average them, and you'll get quite a sensible looking perfusion weighted image. And at this point, that's it. We've got our perfusion image, so we're happy. So the beauty of ASL is the perfusion weighted image is really simple. And so you may well find yourself with data that looks something like this, and the first thing you'll note is that the first image in this sequence looks brighter than the rest of them. We've just learnt about background suppression, so what's been done here is this is a kind of a white paper recommended P-Castle acquisition, but they've used background suppression to remove the background signal that could corrupt the results when we do our label control subtraction. But the first image here is special, and we'll save that for later, so we'll just do the subtraction of the rest of the series. So in this case, I'm going to load it in to a bit of software. All I need to do is load it in and tell it how many post-label delays I've got, and in this case, that's one of them. And I hit subtract, I preview it, and I get my perfusion weighted image. So again, it's simple. This is also here to remind me to tell you that the first thing you should always do with your ASL data is do the subtraction and make sure there's a perfusion weighted image there. So before you go into anything like quantification, make sure it's worked. Always look at your data. Okay, so we kept it simple. That was easy, and we could almost go home here. But let's try and do something a bit more than that. Let's try and get at quantitative measures of perfusion. So we're going to have to start to think now a little bit more about the kinetics of ASL. So as before, we've still got ourselves some ASL data, but we're now aiming for perfusion in absolute units. So we're going to have to take extra steps to account for what's actually happening with our labeled water and think about how we calibrate the process. So from this morning, you'll already be familiar with the idea of tracer kinetics, and you've seen it in other different perfusion cases, and it's all the same underlying mathematical modeling we're doing to understand what is being delivered. But what we can do for ASL is we can try and think about the specific form that the different functions take, and then use that to account for the signal magnitude. So again, tracer kinetics, we've introduced some form of tracer, and we characterize how it behaves in terms of the arterial input function and this residue function, and we say mathematically the signal we measure is the result of a convolution process between these two functions. It's all about delivery and what is present within the voxel that we're measuring within. So breaking that down, our arterial input function is about delivery. So it's going to tell us what is getting to the voxel or the tissue where we're taking our measurement. and relates back to the concentration of the tracer. And for ASL, we know that the process of introducing the tracer is a labeling normally done for the brain in the neck, so somewhere downstream of the organ of interest, which means we're creating what is a fairly well-defined bolus of labeled blood water, of tracer. The main thing we're going to have to account for is that it will take some time to get from where we do our labeling to where we take our measurement. So there's going to be a delay involved, and it's going to decay with T1 while it's getting there. And so what we can start to do now is we can write down our arterial input function. It's defined for us by our labeling process, and we can collect the parameters associated with it. So we need to know something about this, the time it takes for the label to get to the organ of interest, so that we're going to call that our arterial transit time. We need to know something about how much label there was, and that will typically be defined by our label duration, and that's normally something we set in the acquisition. And we need to know something about this T1 decay process. So we've got 3 parameters we're going to have to do something about in our quantification process. And then we also need to say something about the residue function, and we've met this before, and it tells us what happens to the tracer after it has arrived. So it's a slightly unusual function because it's a function of time since arrival. So everything is present at time 0. The little bit of tracer that's arrived at time 0 is all there, but over time it goes somewhere. And maybe it leaves, or maybe it decays. And for ASL, we largely focus on the fact that it decays with T1. And so the standard assumption is to say that everything gets delivered, exchanges rapidly within the tissue, so it's a single well-mixed compartment. Essentially, the water comes in, it goes out of the vasculature into the tissue, and so all of our labeled blood water, all of the labeled water stays there, but it will decay away with the T1 time constant. So again, T1 decay is an important parameter that we're going to need to account for. And so at this point, we can now form the complete kinetics for ASL. We can say it's a combination of the arterial input function and this residue function, the residue function looking something like an exponential decay governed by T1. And so the one extra thing we need to then also note is when did we actually take the measurement within this process? Because we've been delivering a certain amount of labeled blood, and we've let it accumulate, but also we've let it decay. And so this is where our post-label delay or multiple post-label delays come into the picture. What samples have we taken so that we can then go from the samples that we've got back to the magnitude of delivery, our perfusion or cerebral blood flow measurement. I'm going backwards. And actually there's a slight nuance in ASL. So when it comes to the residue function and the combination of the two, we can make 2 slightly subtly different sets of assumptions. And so the very simplest model we can do is to say there is only one T1 value, and it's the same in the blood as well as in the tissue. And we assume that all of the blood water that enters the tissue never leaves. So essentially it stays and decays. And that's the very simplest model we can do. Or we can do what I routinely call the kind of standard model. This is kind of Rick Buxton's work back in '98, which is to say that there's a different T1 in blood and in tissue because they're slightly different environments, and that there's a certain proportion of label that will leave and leak out into the venous vasculature. In practice, it makes fairly small differences to the numbers we get out of the other end. But if you're interested, I do recommend Rick's paper to go and look into some of the details of the model later if you want to. So what this translates to is a certain number of things that you need to know about your data. So you've got some ASL data, but you need to ask some questions of yourself or your friendly physicist or of your scanner, whoever that might be, whatever it might be. So I'm going to assume for today we're looking at pseudo-continuous ASL, but the questions are similar if you're using different variants. You need to know about the time you took your measurement, so the post-labeling delay, or maybe you've got multiple delays. You need to know about your label duration. Importantly, you need to know about the readout, not just because you need to choose a sensible readout, but it will affect the timing. So if you have a 2D readout, it will affect the post-label delay you see. And then you need to make some assumptions about what you're saying in your model. And so this comes back to our T1 parameter and arterial transit time parameter. So so that gets us through kinetics. So we can now describe the time course of the signal in the voxel, we can say something about the relationship between delivery or perfusion and the signal we've measured, but it's still only relative. We still need to know something about the concentration of the tracer. And what we haven't done is introduce something via an injection. What we've done is a labeling process. So how do we say what the concentration is? And this is where we're going to need to know something about the magnetization of arterial blood. So if we go back to our model for a second, we said we need to know something about the arterial input function, and we said about the label duration, which was kind of the time duration of the arterial input function, but we should also say something about its magnitude. And in terms of signal magnitude, this will be related back to the magnetization or the equilibrium magnetization of the arterial blood where we've done the labeling. Because what we've done is we've taken blood with an inherent magnetization, we've applied a label to it, potentially as it's passed through the labeling plane, and it comes out to the other end inverted. And so the magnitude of our arterial input function is directly related to this property of the blood water, but also modulated by how efficient our labeling was. So we've introduced two new parameters that we need to worry about: this equilibrium magnetization of arterial blood and the inversion efficiency. And so we need to try and find a way of calculating this magnetization of arterial blood and doing a calibration step. And in general, we cannot measure this directly, although there are now various techniques that are attempting to do that, but that's not routine yet. What we tend to do at the moment, at least, is we try and get there indirectly via some other tissue or some other element of the organ, particularly the brain that we're imaging. And what we do is we calculate the magnetization of a tissue, of a reference, and then convert that back to the equivalent arterial magnetization. And there are kind of two schools of thought on how you do this, one of which is to say you calculate a magnetization of tissue in every voxel in the organ in the brain that you're imaging, and then you convert that to a local value of magnetization of blood and apply that. Or you can try and do it via reference and say, I'm going to take all of the ventricles, all of CSF, or I'm going to take all of white matter, and I'm going to come up with a single value for the M0 of that tissue and convert that to the single global value of M0. Either way, once you've got that number, that then allows you to take your relative perfusion measure from your kinetics and convert that into absolute perfusion and scale it to get it into the standard units. And if you go and you look at the white paper that now gives a set of recommendations on where to begin with ASL, they will give you a formula that includes all of the steps we've looked at so far. So I'm not going to take you through in detail this formula, but what they've done is they give you the subtraction, the model inversion, and the calibration process, and then they give some suggestions of some of the assumptions you make about some of the other parameters. So, for example, the inversion efficiency. We might want to measure it in some cases, but quite often we're happy to take a standard value for that. So let's actually look at some data now. So go back to the data we had before. So we said we had a series of images, and bingo, there's our calibration image out the front. That is why it's a different intensity. We haven't manipulated it because we wanted a measure of magnetization in the tissue there. And so we know about the label duration, we know about our post-label delay, we know about our readout, and we're going to make some assumptions about the T1, and we're going to make some kind of slightly subtle assumptions about the transit time. And so this is now consistent with what the white paper recommendations might tell us to do. So all this now means is we have to feed in a bit extra information into our quantification step, but all of this is just the standard information we've talked about. We feed in the information about the calibration. So for our first go, we're going to take this voxel-wise approach. We're going to say I'm going to calculate the M0, the magnetization in every tissue voxel, and use that to calibrate each tissue voxel independently. And if I follow that through, I finally get myself my perfusion image in my standard units. And one of the caveats with taking the voxel-wise approach is it tends to have issues around the edge of the brain where we've got partial volume effects. But we can do a little quick correction for that and get ourselves what is quite a nice and standard appearing perfusion image. Interestingly, if I took the alternative calibration approach, so here if we take the ventricles and do a calculation of a single global equilibrium magnetization value, then actually reassuringly I get pretty much the same answer. And so the nice thing about having options here is not so much that it's important which one you select, but you can choose the one that's right for the scenario you're in. So if you're looking at a population where you can't realistically quantify something from the ventricles because you've got blood leaking in there, then you've got a backup option and vice versa. And we can go a bit further. We can do the same thing, but now we can do it with multiple post-labeling delays. So we've sampled different points on our kinetic process, our kinetic signal curve. We can feed the data in. We just have to be a little bit careful. We understand what order it's come in. But essentially, it's exactly the same process as before. And reassuringly, in a single subject, we get very similar answers. But what we now get with our multiple post-labeling delay acquisition is it also gives us a measure of the transit time, the arterial transit time parameter. So rather than having to make a strict assumption about that value, we can now control for its variability across the brain. And we see that there is a certain amount of variability in this case, and it tends to be, you get longer transit times towards the back of the brain. Okay, so we've talked about quantification, we talked about kinetics and calibration. Now a little bit of time just to think about some of the next steps. So if this is part of a larger study, and so you've got to then take this up to group analysis, there's just a couple of things that's worth being aware of. And really it comes down to consistent geometry and consistent intensity. So how do we normalize individual subjects so that we can compare them in a common space and ensure that we can compare them consistently between each other in terms of intensity? Consistent geometry is just the classic registration process, and the caveat with ASL is that because you've got a relatively low resolution image and potentially limited SNR to start with, that first step of registering your perfusion image to your structural image, which you then use typically to get to your standard space or your template space, can be a challenge, but you can exploit the advantage that you get good grey matter, white matter contrast in perfusion images to do this process. And so we've done spatial normalization. We also need to worry about intensity normalization. Well, in practice, we shouldn't need to because we're generating quantitative maps. So in principle, we should be able to compare individuals based on a fully quantitative measure of perfusion. And that would allow us to then set consistent thresholds. So if we were looking for ischemia, we should be looking for 20 mil per 100 gram per minute. And this is fine as long as we've got calibration information. But the one big caveat with this is that perfusion varies quite a lot between individuals. And so in practice, there's a source of variability in a group here that is not easy to control for. There's just natural variability. And that will, in effect, add extra noise to your group study. So there are cases where you might want to intensity normalize, which then avoids the need for calibration, to allow you to at least look at local variations and local differences. But you have to be a little bit careful that you have a consistent reference for doing that. And then, of course, you've thrown away some of the advantages of having a global absolute measure because you can no longer detect global changes. So, for example, if you're looking at a pharmacological study, you've thrown away the ability to see whether the drug has a global change in perfusion. So the last few minutes, I just wanted to look at a couple of more advanced quantification issues, one of which is distortion correction, which has become widely used for sort of fMRI and bold studies, but applies to ASL just as much. And the trick here is we can use exactly the same processes they do in fMRI and in diffusion, which is to say, if I can get some sort of field map, I can then calculate what the distortion was. And so a classic way of doing this, and a good one for ASL, is if you haven't got a field map from somewhere else, you can't nick it from your diffusion scan, is to do your calibration image twice with different phase encoding, and then combine it together in using one of the kind of standard tools for this. So in this case, we've got anterior-posterior and posterior-anterior encoding, and you can see quite large distortion effects around kind of where we'd expect them to be, and we can correct for that, but we have to be particularly careful with ASL because we are doing absolute quantification, that we correct not just for the spatial distortion, but also the intensity changes associated with that. And so this is returning to our example data. This data set had that calibration image with the two different phase encodings. And again, we get ourselves a perfusion image where we've managed to correct. And so we're getting closer to something that's now very easily comparable across individuals because we've corrected for different sources of distortion and variability. So our final topic today is to think about another issue that we can easily overlook for ASL, and that's related back to the resolution that we're sampling at. But it feeds back into this issue of we're doing absolute quantification. So, because we're doing relatively low resolution, we're going to get partial volume effects between grey matter and white matter. And this is important because white matter perfusion is a lot lower than grey matter perfusion. That is why we get the images looking the way we do. And so really, we potentially want to be able to separate out these two effects. So we should ask ourselves the question, how much does it matter? So if we think about a typical ASL resolution and the kind of standard cortical thickness, you will find that at ASL resolution, there are very few voxels of pure grey matter. So therefore, it's very hard to quote grey matter perfusion from ASL. It's really a mixture. And that leads to this kind of interesting result, which is if you compare a perfusion image on the right here with something that's just based on partial volume estimates of grey matter, down sampled to the same resolution, you get a very similar looking image. So the message here is if you look at a perfusion image from ASL, what you're looking at is partial volume of grey matter with perfusion modulated on top. So can we pull that modulation back out? And in essence, what we want to be able to do to pull that modulation back out is to separate the two components. And we're going to separate them using two different features, potentially. One of which is we will have separate estimates of the partial volume effects, hopefully from a structural image, and this is where distortion correction is important. And then if we've got multiple post-label delays, we can use some of the timing information to leverage and help us further. And we can do this, and I'm not going to go into the details of how we do this today because of time, but I'm going to take the data set we were looking at a moment ago, I'm going to add in my structural images, and now I can potentially separate out grey matter and white matter perfusion, and the big question then remains, well, what does that mean and what have we achieved with this? So actually, we've hopefully got grey matter perfusion, but there's still a few questions about how we start to interpret something that we've not had before. So the last thing to say is I've shown you FSL tools. There are other tools. Pick the one that suits your platform and suits what you're most familiar with. If you want to know more, there are various places you can go and various books to look at. Thank you very much.